Hello, a warm welcome to all the dear learners. Today I am going to discuss about the T cells from its origin to its functions. Before starting today's lecture, I would like to give you a brief introduction about the T cells. We know that white blood cells are involved in the defense mechanism of the host. Here, the blood cells originate from the bone marrow, and the cells which are responsible for synthesizing these blood cells are called as the stem cells, or we can say pluripotent hematopoietic stem cells or totipotential stem cells. And it is divided into two types, that is myeloid stem cells and lymphoid stem cells. All the cells except the lymphocytes originate from the myeloid stem cells, and the lymphocytes originate from lymphoid stem cells. Lymphoid stem cells are further divided into two types, that is the B lymphocytes and the T lymphocytes. And we know that lymphocytes are a type of white blood cells under the category of agranulocytes. Rest all the blood cells, including the white blood cells, are synthesized by the myeloid stem cells. And in this is today we are going to discuss is about the T cells. The other cell is the B cell. So T cells, if you are aware of the immune system, so T cells constitute our cell-mediated immunity, and it plays a crucial role in defense mechanism and providing immunity to the host cells against different type of pathogenic infections. It is necessary to understand its complex functions because it gets activated through a series of interactions between the pathogen and how it reacts or responds against those pathogens. So today we will discuss about its origin as well as the way it functions through a series of chemical reactions. So let's start today's lecture. So if we give, as already I have told you, that lymphocytes are basically the cells which originate from the lymphoid progenitor cells in the bone marrow. Progenitor cells are the precursor cells, which it is a process of a type of hematopoiesis, and that too, that is also called as the leukopoiesis, through which these white blood cells are formed, and it is from the lymphoid origin. And after formation, they migrate to the thymus. We know that B cells usually mature after the formation. They mature in different parts of the body. If they mature in the bone marrow, they are called as B cells, and if they mature in the thymus, they are called as the T cells. Now, on the T cells, there are different type of receptors present, which is mostly the CD3, CD28, and T cell receptor protein on their surface. And they constitute 65 to 80% of the circulating lymphocytes. Within lymph nodes, they are located in the inner subcortical regions. and their lifespan of T cells is longer in comparison to other cells. Now, one of the important functions that basically, if you see all other immune cells, either it is phagocytic cells or other antibodies, so they react or interact with antigens directly and destroys them. But T cells recognizes antigens only when they are presented on antigen presenting cells like macrophages, dendritic cells, and B cells. When and second, when they displayed on major histocompatibility complex protein, and only when they are peptide antigen, that is proteinous in nature. Next important functions, next important role of T cells are that T cell precursors or lymphoid or progenitor cells arise from bone marrow and they reach thymus where they are called as thymocytes and mature here under the influence of thymic hormones and then released into the periphery. During maturation with thymus, T cells acquire receptors and undergo thymic education before being released to into periphery. So, if I summarize it, basically the T cells, when before getting released into the circulation, they first mature in thymus or lymph nodes, and then they get differentiated. Means that there is a called as a selection theory that how only those cells will be circulated which reacts with the foreign antigens and not with the self antigens. So we will see what is the different stages of differentiation. So prior entering the thymus, the cells lack antigen receptors and CD proteins. So before maturation, they are just simply uh, normal cells. They are not getting, they have not acquired the different receptors which differentiate, helps differentiate between the different T cells on the surface, and these are called as the double negative cells. Now, after that, there is a rearrangement of T cell receptor gene occurs and they differentiate into T cells that can express antigen receptors and various CD proteins, that is namely CD3 plus, CD4 plus, CD8 plus. So double negative cells become double positive cells. That is, they acquire T cell receptors as well as CD proteins on their surface, and hence from double negative to they become convert themselves into double positive cells. Now here cells here CD4 plus and CD differentiate cells that do not react with self-MHC proteins are killed. That is, there is a positive selections. Here, what happens that DP cells undergo thymic education at this stage, that is positive to negative selections. Now, negative selection occurs. Here, CD4 plus and CD8 plus cells bearing antigen receptors for self proteins are killed. As I already told you, that only there is a process, this is the procedure in which there is a memory, or we can say there is a change in the processing of the differentiation in the T cells, that on such cells which have receptors CD4 plus and CD8 plus, which uh, receptor for self proteins are killed, that it will not react. Those who are able to react with the cell proteins are killed, and it results in tolerance to own cell proteins, that is the self tolerance, and this self tolerance prevents the autoimmune reaction. Autoimmune reaction in brief, if I would like to say that Such conditions develops when immune system in uh, in addition to destroying foreign cells, they start destroying their own self cells, and such conditions are called as autoimmune reactions. So it is necessary that such cells are needed to be eliminated during the processing of the T cells, so that they do not react with their own self antigens. Means that they should not recognize the host cell itself. Otherwise, it will start destroying the own harmless cells, and it will lead to some autoimmune immune reactions. So here what happens, that it results to the, this is also called as the process is called as the self tolerance, and this step is called as the negative selection. So first it is double positive, and then it converts into negative selection theory. Negative selection occurs. Now selection of double positive cells by MHC class 1 or MHC class 2 molecules to become CD plus and CD4 plus react respectively. That is, they become single positive cells. If I explain in more detail, here what happens, that if when the cell differentiate into T cells that express antigen receptors, that is T cell receptors and various CD proteins, they become double positive cells. Now, double positive cells undergo thymic education at this stage. That is, there is one positive again, positive selection and negative selection. So here in negative selection, as I have already told, that where CD4 plus cells and CD8 plus cells bearing antigen receptors and TCRs that do not react with self MHC proteins are killed. That is positive selection of T cells whose TCR reaction react with self MHC proteins. And then what happens? There is a negative selection occurs. Here what happens? Here CD4 plus and CD8 plus cell bearing antigen receptors for cell proteins are killed. That is negative selection mediated by signal transduction through T cell receptors, leading to induction of apoptosis and eventual clonal deletion. The removal of these self-reactive cells through the process of negative selection results in tolerance to on proteins, and that is called as self-tolerance, and this self-tolerance prevents autoimmune reactions. After that, TCR expression increases and selection of double positive cells by MHC class 1 or MHC class 2 molecules to become CD8 plus and CD4 plus respectively. That is, they become single positive cells. Now, through positive and negative selection, T cells are selected for their ability to react both with foreign antigen and self MHC proteins. Both these features are required for effective immune response by the cells. These cells leave the thymus and reach peripheral lymphoid organ, where it gets activated on interacting with foreign antigen. So we can say in detail that first there is a double positive, then there is negative selection, where where T cells reacting with own antigens are eliminated, and again there is single positive reaction. Single positive reactions means that two T cell subsets will form, one having CD4 plus cells and one having CD8 plus, and thus either they will acquire CD4 plus if they are, or they will acquire CD8 plus along with T cell receptors, and these selection of by MHC class 1 or MHC class 2 molecules. Now after that, here again, if I can say that earlier from the stem cells or development or maturation of T cells occur from the bone marrow, there is they are basically, they are double negative, that they neither possess CD4, CD8 or T cell receptor on the surface, and again they become double positive. Double positive means that they have both the receptors. CD4 plus is also there, CD8 plus is there, and then T cell receptors are there. After that, there is one positive selection. Positive selection involves the survival of cells with TCR, T cell receptor that react with self MHC proteins. Here, remember that not with the self uh, proteins. It is the self MHC proteins, that is MHC class 1 and MHC class 2. So that is called as the positive selection and death of cells with T cell receptors that do not react with cell MHC. Remember that those T cell receptors will be eliminated, or they will lyse, which do not react with self MHC proteins. Now this is called as the positive selection. Now again this CD4 plus CD8 plus and T cell receptors acquired through the positive selection further goes into the negative selection, which involves the death of cells with T cell receptors that recognize self antigens, and those cells are eliminated, and only those cells survive which have CD4 plus, CD8 plus and T cell receptors, and further which again gets differentiated into either CD4 plus T cell receptor cells or CD8 plus receptor cells and TCRs. In other words, if I would like to say that it will be CD8 plus, CD4 minus, TCR plus. One cell, single positive, and other is CD4 plus, CD8 minus, and T cell receptors, single positive cells. So I hope you'd be understanding the concept of the formation of single positive cells. Now here, through the diagrammatic representation, I can show you that here you can see here that this is double negative. Okay, then it converts into double positive cells, that is a CD4 plus and CD8 plus. In step 3, what happens, that MHC induced negative and positive selections occurs here, and further it develops into single positive cells, that is CD4 plus, CD8 minus, or CD8 plus and CD4 minus cells. Next, if we see what are the receptors which are acquired on T cells during maturation are, that is T cell receptor CD3 and CD28 present in all mature T cells. In addition to CD4 and CD8 selections, these are the common receptors which are present on all the mature T cells. So what are their functions? If we see there, then T cell receptors, it is an antigen binding receptor. Then CD3 complex is the signal transduction of T cell receptor lineage specific marker. Then CD2, CD5, and CD7, that is the lineage specific marker. Again, CD4 is subset specific marker mainly on helper T cells, and it interacts with class 2 MHC proteins. That CD8, which is a subset specific marker mainly on cytotoxic T cells, interleukin with class 1 MHC protein. Then CD40 ligand, action specific marker, delivers contact mediated help to B cells. Then interleukin 2 receptor class 2 MHC protein, transferrin receptors CD25, CD29, CD54 and CD69, that are other activation specific marker, and interleukin 1 receptor, interleukin 6 tumor necrosis factor alpha receptor, that those are the other type of cytokine receptors. Again, in addition to that, Fc receptor, that is the immunoglobulin binding, then LFA1 and ICAM1, that is the cell-cell adhesion molecules. These are some of the receptors which are present on the T cells. Now coming to the next part, that is what are the T cell receptors. If we see here, so the antigen receptors present on T cells is called as the T cell receptor, and all mature T cells have T cell receptors present. And this is also, there are T cell receptors are of two types, that is T cell receptors with 2 beta polypeptide chain. 95% of T cells are of this type, and T cell receptors with gamma delta polypeptide chain, 5% of T cells. Now here, what is the genetic component of T cell receptor? So T cell receptors belong to immunoglobulin supergene family, which includes immunoglobulin, T cell receptors, and human leukocyte antigen protein. And therefore, their basic structure and genetic configuration is similar to immunoglobulin. So, their structure and genetic configuration is almost similar to immunoglobulin molecule, and gene that code for them are found by the rearrangement. There are four segments of TCR gene. One is V for stands for variable, D diversity, J joining, and C constant. In immunoglobulin also these genes are present, and these segments rearrange to form T cell receptor diversity, and they are mostly responsible for the diversity, means that the variations in the T cell receptors, and more than 10 to the, and because of their recombination, all these four genes, they are able to make more than 10 to the power 7 types of T cell receptors are present. The variable regions has, again in them there are hyper variable domains, and the enzyme which that catalyzes the gene rearrangement are also present, that is RAG1 and RAG2, which is the short form for recombinase activating gene. Next, coming to the next slide. So this is, you can see through the diagrammatic representation that alpha chain here, and T cell, an ideal figure image of a T cell receptor which has coded by the variable and constant region. This is the transmembrane region where their stem is attached, which is attached to the through the disulfide bridge, and inside it there is a cytoplasmic tail. Now, what are some of the important functions of T cell receptors? Before that, some of the important difference between T cell receptors and immunoglobulin, that T cell receptors can bind antigen only in the presence of MHC protein, whereas immunoglobulin can bind free antigen. Then it is present on T cells, whereas immunoglobulin are present on B cells, and they are also produced and secreted by plasma cells. Then T cell diversity depends on joining of V, D, J, and C segments, and combining effect of alpha beta polypeptide chain, and does not depend on mutations, whereas B cell is that present on B cell, and they are also produced and secreted by plasma cells. And antibody diversity depends on rearrangement of VDJC segment, whereas in the T cells they are dependent on joining. And here there is a antibody diversity depends on rearrangement of VDJ and C segment. Then combining of different light and heavy chains, whereas here alpha and beta polypeptide chains are present, and on one hand we see that it does not, the T cells does not depend on mutation formation, whereas B cells immunoglobulin forms depends on mutations. So here some of the important differentiation between the T cell receptors and immunoglobulin we can see easily. Now here how the activation of T cells takes place. So T cell recognizes peptide antigen only when presented by antigen presenting cells, as we have already discussed, that is the macrophages or dendritic cells in association with MHC proteins. That is, T cells are MHC restricted. Next here, only on the basic of MHC restriction and antigen presenting cells, they can be processed. Now, how the mechanism of antigen processing by antigen presenting cells takes place. Now, in these functions, we can say that either through extracellular antigen or exogenous antigen, through it can take place, or through it may take through the intracellular or endogenous antigens. Now here one more important topic, that is called as the super antigens. So what are the super antigens? So super antigens are those antigens which do not require the processing through antigen presenting cells. It does not require the process of antigen presenting cells. So before going to the super antigen, let me explain first the what are the exogenous antigens and endogenous antigens. So extracellular antigen or exogenous antigens are as toxins, microbes, then growing in the intracellular vesicles, protein or vaccine. Here, the processing of antigen takes place within the endosomes where it is degraded by two peptide of different sizes that can bind to MHC class II molecules, which are then recognized by T cell receptors. Whereas the intracellular or endogenous antigens such as viral proteins and bacterial that replicate in cytosols. So endogenous antigens are endosomes and here exogenous antigens are in the cytosols. Here the antigen is cleaved enzymatically by proteosomes to peptide fragments which bind to MHC class 1 molecule. So here we can see that how the important difference between the endogenous antigen and exogenous antigens. Now coming to the super antigens. So some antigens are termed as super antigens can activate T cells without requiring antigen processing. The super antigens binds as you can see here, the binds simultaneously to the external surface of MHC class II molecules on antigen presenting cells and to the external site of T cell receptor. These bindings not a part of typical MHC antigen binding site complex. A result of this binding, they activate and expand large percentage of T cells leading to severe systemic compositions. Now, super antigens include bacterial exotoxins such as staphylococcal exotoxin or some retroviral antigens. So here they do not follow the classical antigen presenting cells through which T cells are activated. They directly binds to the external surface of MHC class II molecules of antigen presenting cells and to the external site of T cell receptors. So this is the basic difference between the exogenous, endogenous and super antigens. Next is the process is that the mature T cells that leave the thymus and have not encountered the antigen are said to be the naive T cells. Naive T cells are in constant circulation via blood streams and lymphatics. They recirculate till they encounter antigen or a dye. This recirculation maximizes the chance to encounter antigen displayed on antigen presenting cells, which is a requirement to activate naive T cells. Until and unless they do not encounter an antigen, they remain as naive T cells. Now, how it gets activated? So, foreign antigen brought to secondary lymphoid organs by dendritic cells. Dendritic cells and antigen presenting cells release cytokines, which functions as signals to infections. This brings already circulating naive T and B cells into proximity with antigen presenting cells. B cells with B cell receptors take up antigen via receptor mediated endocytosis. T cells recognizes antigen presenting bearing foreign antigens, which is cleaved into small peptides that associate with class II MHC proteins and transported to cell surface of macrophages and T cell activation occurs. So this is the mechanism how naive T cells are activated into CD4 plus T cells. Now here you can see here through the diagrammatic representation that how the interaction between the antigen presenting cells and T cell receptors and other cell surface receptors occurs. Now here activation of CD4 T cells requires at least two signals. First signal is the interaction between T cell receptor and antigen MHC class II complex, as we have already discussed. While this interaction occurs, CD4 proteins on the surface of helper T cells also interact with class 2 MHC protein. Other proteins that interact to help stabilize the contact between T cells and proteins, antigen presenting cells are LFA protein. There is an LFA protein on T cells which bind to ICAM protein on antigen presenting cells, which helps in proper cell to cell adhesions. Then the second signal is a co-stimulatory signal. The second signal is a co-stimulatory signal, that is interaction between CD28 present on helper T cells and B7 proteins on antigen presenting cells. If this co-stimulatory signal is not present, the T cells become anergic. Now, what is the anergic T cells? So, anergic T cells are those T cells which which remains viable, means that it is not it is functional, it is a functional, but it is refractory to stimulation by antigen, so it does not get activated, and therefore no immune response. It can be long lived, persisting for weeks and months. So that is the importance of co-stimulatory signal. So if even the MHC class 2 proteins on MHC class 2 proteins and T cell receptors on the T cells interact, but if co-stimulatory signals, that is the B7 and CD28 do not react, it will not get activated. And the signal of recognition of antigen is sent into the T cell through CD3, which results in activation of phosphokinase and phospholipase, which open the calcium ion channels, and calcium enters into the T cell and activates calcineurin, which activates gene which encodes IL-2 receptors, and IL-2 and IL-2. The IL-2, also called as T cell growth factor, produced by the activated T cell, can act on same T cell autocrine function or neighboring cells, that is paracrine cell functions, helper T cells or cytotoxic T cells. Some of these activated T cells can proliferate into memory T cells, which retain the memory of antigenic stimulation and brings about secondary immune response. So that is a separate topic, that is how the primary and secondary immune response takes place. So here again, after the importance of co-stimulatory signals, that is CD28 and B7, here the function of interleukin 2, which is also called as the T cell growth factor, also holds an equally important in the function of the both helper T cells and cytotoxic T cells. Now, certain mitogens also activate T cells. Some of the important are phytohemagglutinin and concanavalin A. If T cells are activated continuously, it may result in harmful reactions. So if there is a continuous stimulation persist, again it will become harmful, and so it needs to be checked that only there is a need for a check on the stimulation of T cells, and this regulation is brought by a surface protein called as the CTLA4, which appears on helper T cells and interact with B7 on antigen presenting cells. This interaction suppresses T cell activation. So it acts as a regulatory mechanism. Now, we already know that there are two subsets, two major types divided through single positive selection, that is CD4 plus lymphocytes and CD8 plus lymphocytes. So CD4 lymphocytes, they are also known as the helper T cells, and they bring the following functions. The major role of helper T cells is very crucial even in the activation of B cells as well as CD8 plus or cytotoxic T cells. So they activate antigen specific helper T cells to produce a clone of these cells. And as we discussed, that a very important cytokine, that is the interleukin 2, which mediates this function. Again, they help CD8 T cells to become activated cytotoxic T cells, and the cytokine responsible is interleukin 2. Then third is the activation of B cells to develop into antibody producing plasma cells. And here the cytokine which mediates the function is interleukin 4 and interleukin 5, and in activation of macrophages, which is mediated by interferon gamma. Now, these are the some of the important functions. Now, CD4 lymphocytes, which makes up of 65% of peripheral T cells, it predominates in thymic medulla, uh, tonsils of thymic medulla and tonsils and in blood. Now, CD4 lymphocytes have two subpopulation of CD4 cells. Again, it is divided into two types, subtypes. One is T helper 1 cells and T helper 2 cells. Above functions are performed by these cells. Now, what are the origin of T helper 1 cells and T helper 2 cells? So, we can say that T helper 1 cells T helper 1 cells and T helper 2 cells, microorganisms are ingested by macrophages and some unknown. So microorganisms are ingested by macrophages and some unknown cells. Example, eosinophil, mast cells in parasitic infections. Microorganisms ingested by macrophages stimulate production of interleukin 12, which stimulates the naive T0 to become T helper 1 cells to produce gamma interferon and interleukin 2. And microorganism ingested by unknown cell induces the production of interleukin 4, which stimulates naive Th0 to become T helper 2 cells that produce interleukin and interleukin 5. Interleukin 10 is also produced by T helper 2 cells. So here you can see here some of the important functions of T helper 1. So basically we can say that those micro or antigen presenting cells which is constituted by macrophages are it stimulates the Th0 to become T helper 1, and whereas some unknown antigen presenting cells, for example, eosinophil or mast cells in parasitic infections, they help Th0 or naive T cells to become T helper 2 cells, and that become to produce interleukin 4 and interleukin 5 and also interleukin 10. So here some of the important regulation are regarding the T helper 1 and T helper 2 cells in so here we can say that the T helper 1 and T helper 2 cells, how it is formed. Then next important thing about the T helper 1 and the regulation, and interleukin 12 produced by 12 produced by macrophages increases the number of T helper 1 cells, thereby enhancing delayed hypersensitivity response, and gamma interferon produced by T helper 1 cell inhibit the production of T helper 2 cells, and interleukin 10 produced by T helper 2 cell inhibits the production of interleukin 12 by the macrophages. So how they have the effect on each other. So one secretion or one cytokine secretion from one T cells will inhibit the production or activation of other T cells, and so that their functions remain specific. Now CD8 lymphocytes, also known as cytotoxic T cells, constitute about 35% of peripheral T cells predominant in bone marrow and gut. Now, what is the functions of CD8 lymphocytes or cytotoxic T cells? Here they kill virus infected cells, tumor cells and allograft cells by either two mechanisms: release of perforins, which destroy cell membrane, and through apoptosis. Here, if we compare the T helper 1 and T helper 2 cells, produces interleukin 2 and interferon gamma is produced by T helper 1 cells, whereas interleukin 4, 5, 6, and 10 is produced by T helper 2. Enhances cell mediated immunity and delayed hypersensitivity by T helper 1 but not by T helper 2. Antibody production is enhanced by T helper 2 cells but not by T helper 1. Stimulated by interleukin 12, as we have already discussed it, that it is by T helper 1, and stimulation of by interleukin 4, it is of T helper 2 cells. And functions of T cells are divided into two main category, that is regulatory functions, which is performed by helper T cells, and effector functions, which is performed by cytotoxic T cells, which we will discuss in our next lecture. So today till this we conclude our today's lecture, and I hope that this would have been beneficial for you to in understanding the mechanism and functions of T cells. If you do like my lecture, my teaching, please like and subscribe, which will keep me motivated to bring more information regarding the medical's different parts of the microbiology, as well as some of the important topics related to it. Thank you, everyone. Have a good nice day. Thank you.